Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Excretory Products and Their Elimination. Let's move on to the process of urine formation. But before we dwell into that, we have to just recollect the parts of our nephron, because we know that within the kidney a lot of nephrons are filtering units, or millions of nephrons are there. They are filtering out the waste from the blood, right? So where is this mainly happening? In the glomerulus. You know, glomerulus means what? The tuft of capillaries forming inside the Bowman's capsule. It is a branch of the artery that is forming it. So when we think about that, we can say that, suppose, so first the branch of artery is coming, what is it called? That is called the arteriole, right? This arteriole is called the afferent arteriole. Then it comes and branches inside to form many thin-walled capillaries, and then it goes out as efferent arteriole. But there is a difference in the size of this afferent and efferent arteriole. Afferent is larger, efferent is smaller. So from a larger vessel blood is coming, and it is then entering into fine tubules, and it goes out by a thin tubule. So naturally what is happening? The tension increases. So the blood pressure inside the glomerulus is little high. And we know that the blood is filtered here into the space and then to the Bowman's capsule. Suppose if I enlarge, this is the blood vessel, okay, blood vessel of this glomerulus, and then it is coming down to the Bowman's capsule. So this is the Bowman's capsule, and in between there is a space. So the filtrate has to come through different layers. First it has to cross the endothelium of the blood capillary, glomerular blood capillary endothelium. Then this part is called the basement substance, that is a substance in between these two layers. Then comes the epithelium of this Bowman's capsule. Now in this endothelium of the glomerular blood capillaries, there are lots of holes in it. That is why the filtration is happening like ultrafiltration. At the same time, the endoepithelium of the Bowman's capsule also has cells called podocytes. These cells have slit pores in between. So through that the filtrate can come inside. So almost all contents of the blood except the blood cells and protein will come inside the Bowman's capsule in the form of a filtrate. At this time we don't call it as urine, it is called a filtrate. So since everything is filtered out except for the cells and the blood cells and protein, we call it as ultrafiltration. Now, NEET point of view, it is better to know about the types of cells found at different parts of the nephron. So, you know, nephron starts with the Bowman's capsule, and from the Bowman's capsule, it enters into the first convoluted part, that is called a proximal convoluted tubule or PCT. So, in the PCT, basically, cuboidal epithelium are found. You know, epithelial cells can be squamous epithelium, very thin. Then it can be cuboidal epithelium, means cube-like, which are mainly supportive. Then columnar epithelium, which are tall, column-like. They are also mainly for secretion. And finally, glandular epithelium, which can be a modification of the squamous epithelium for secretion. So in this case, cuboidal, or if it is columnar, they can have cilia sometimes. Then we call it as ciliated cuboidal or ciliated columnar accordingly. In this case, PCT has cuboidal epithelium, but it has lot of villi, means microvilli, that is called a ciliated cuboidal epithelium, and it gives a brush border appearance. This is mainly for increasing the surface area, because we know that villi serve the function of increasing the surface area. Then when we go to the next part, which is the next part, the U-shaped coiled structure called a loop of Henle. In loop of Henle, the part which is coming down is the descending limb, and the one which is going up is the ascending limb. In the ascending limb, squamous epithelium is found, whereas in the descending limb you will find cuboidal and some columnar epithelial cells. In the Henle's loop, the reabsorption is not similar to that in the PCT, where the maximum reabsorption happens. Then DCT, distal convoluted tubule. There we have again cuboidal cells which are supportive also in nature, but here the difference is they are not having brush border appearance because their number of microvilli are less. And finally, you know that many DCT from different nephrons come and open into the collecting duct. But these collecting ducts also eventually join together, a few of them, forming duct of Bellini. They will be opening out into the pelvis, releasing the urine. Now we will move on to the process of urine formation. Urine formation actually involves 3 steps. The first step is glomerular filtration, or we call it as ultrafiltration. Means majority of the content of the blood is filtered out. Then many of the materials are needed for the body, so we need to take them back. That is the second step, that is called a selective reabsorption. We are not reabsorbing everything, selectively we are reabsorbing. That's why we use the word selective reabsorption. And what is the third step? Tubular secretion. It's not only we are absorbing things from the filtrate, but we are adding some ions and substances into it also for maintaining the osmolarity. Right? So let's learn one by one. First is glomerular filtration or ultrafiltration. As the name indicates, where is it happening? It is happening in the glomerulus, or rather the Malpighian body. What is Malpighian body? The glomerulus along with the Bowman's capsule. And as I mentioned now, that is, it is passed, the blood is coming under the pressure, because from the afferent arteriole, when it enters into capillaries, it is under high pressure. As a result, all the contents except some proteins and fats and some blood cells, everything will come down. That is, all the constituents of plasma come down through 3 layers. Endothelium of the Bowman's capsule, which has many pores in it. Second, basement substance, and the podocytes are forming pore slits through which the blood will enter into the, filtrate will enter into the Bowman's capsule. So, since almost all contents are filtered out so finely, we call it as ultrafiltration. So, glomerular filtration is also called ultrafiltration. Why? Can be a question. Now, when it is filtered out, we know that during the filtration process, almost 1100 to 1200 milliliter of blood passes through the kidney per minute, both the kidneys per minute. That is almost 1/5 of the total blood pumped by the ventricle. So in 1 minute. So from that 1200 milliliter of the blood, how much filtrate is forming? It is 125 milliliter of filtrate is forming per minute. This value is called the GFR. GFR stands for glomerular filtration rate. That is amount of filtrate formed by glomerular filtration per minute. That is 125 milliliter per minute. But if you calculate that, we will get 180 liters per day. 180 liters means it's a huge number, because we know usually we buy the water bottles, drinking water, that can be 1.5 liters water bottle, right? Usually we buy that 1.5. So the amount produced is 180 liters per day. That is equivalent to some syntax tank we use for storing water on the terrace or somewhere. That is almost 200 liters what we buy, or big ones are also there, but on an average 201 is quite big. So 200 liters, you imagine that much urine or filtrate is forming per day, and if we start excreting out that, we will not have time for anything else than urinating, right? So many of the contents or this much water we cannot lose, so we reabsorb them. The reabsorption is happening to a greater extent that almost 99% is reabsorbed back. So from the filtrate, 99% of water and other substances are taken back. As a result, finally, usually a female will be excreting 1.2 liters of water per day. On an average, a male excretes only 1.5 liters of water per day. So that means from 180 liters of filtrate, we are making only 1.5 liters of urine in our body, which is only 1% of what is filtered out. So reabsorption is very significant here, because when initial filtrate is forming, it has got so many things in it. Mainly urea with the water is there. That is our main waste to be excreted out. Then glucose and amino acids are there in the blood. They are the digestive products. With the great efforts, we removed them from the food and we absorbed it. Now we have to use it for our use. We cannot just excrete it out. So we need to take them back. Then what is there? the ions are there, sodium is there, potassium is there. Okay, so like ions are there, then some amount of uric acid, which is another nitrogenous waste, is there, and some creatinine. All these need to be excreted out. But we need to take back lot of water, we need to take back some ions which are needed for our body. Of course, we need to take back the nutrients. But how will we take them back? It is by either active absorption or by passive absorption. Active means what? We need to spend energy to bring them, because already we have enough, but from lower concentration again we are bringing to the higher concentration, so that is active absorption. Whereas if it is from higher to low, it will go just by osmosis or diffusion, that is called a passive. So if energy is used, it is active, that is ATP is used, but without the use of ATP, it is going from high to low, that is called a passive absorption. So either of this will happen during reabsorption. Now let's look at the selective reabsorption process. I told 99% of the ingredients of the filtrate are being reabsorbed at this stage. So in this regard, there are three regions which are mainly involved in reabsorption. The nephron part, first part is PCT. So PCT is the proximal convoluted tubule where the maximum reabsorption happens. That is almost 65 to 80% of the reabsorption happens in the PCT itself. Where what all things are taken back? Glucose and amino acid, obviously we cannot lose them, they are the nutrients. Then some electrolytes and water are reabsorbed, and this is called an obligatory reabsorption, because there is no choice the body has. It has to reabsorb all these things to get back to the body. We don't want to lose them. And at the same time, the secretion happens. This secretion part is mainly, that is the third part actually, tubular secretion. The third step is mainly for maintaining the osmolarity, that is pH and ionic balance of the body. That is why the water level is kept okay for our body. We should not hold more water, or we will end up in swelling called edema, or we should not lose much water, which will end up in dehydration. Both are not good for us, so we need to maintain a proper balance of water. For that, pH and ionic balances are also important. So for that, we secrete a few substances into the PCT from the medullary interstitium, means nephron is there in the medulla region. So behind the nephron, the cells are there. That part is called medullary interstitial cells. And then the blood capillaries, peritubular capillaries we talked about in the previous video, that is also there. So from there, some substances are coming into the filtrate or into the PCT. Which are substances are added? They are not substances, they are ions. Hydrogen ions, H+, potassium ions and ammonium ions. But at the same time, for maintaining osmolarity, we are taking back HCO3- or carbonate ion to it. To maintain the secretion level, we are reabsorbing this bicarbonate ions. Now coming to loop of Henle, as I mentioned earlier, loop of Henle has minimum reabsorption happening, but its function is different. It is mainly for concentrating the urine. It has to keep the medullary interstitium highly osmotic, means the osmolarity should be high there. This is maintained by our loop of Henle. So loop of Henle has two limbs, ascending and descending. So first descending limb, it is permeable to water but impermeable to electrolytes. Whereas ascending limb is impermeable to water but permeable to sodium ions. So this will maintain a high osmolarity, or we can say very salty nature of the medullary interstitium or the medulla region, which will help in the formation of concentrated urine, or which will help us to reabsorb maximum water and ions from it. Now coming to the third part, that is DCT. DCT is doing conditional reabsorption. Here it was obligatory reabsorption. The body had no choice, it had to reabsorb. But almost 80% reabsorbed. Now whatever is left, if the body wants them back only, it has to be reabsorbed. So that is called the conditional reabsorptions. It can happen, cannot happen. So which are substances are reabsorbed? Again, sodium is reabsorbed, water is reabsorbed. Now nutrients, because they are all already taken back. Now only sodium ions and water, depending upon if more water is going out of the body, we take back, right? If enough excess water, enough water is not going out, means we will not take back. It's like that. Then again, there also the secretion remains the same, H+, K+ and ammonium ions. To maintain the osmolarity, the secretion is happening, but for adjusting that, we will reabsorb bicarbonate also from the DCT. Now finally coming to collecting duct. Collecting duct, there also water is largely reabsorbed in the collecting duct, so that towards the end of the collecting duct we get maximum concentrated urine, and to maintain that osmolarity in that region, urea is also being reabsorbed at the collecting duct part. So urea is mainly reabsorbed from the collecting duct, whereas sodium is from the ascending limb of the Henle's loop, and later reabsorption happens in the sodium. Reabsorption is mainly in the DCT depending upon the need of the body. Hope you like all the lessons I teach. If you find it useful, please share with all your friends, give it a like, and if you haven't subscribed to my channel, please subscribe. Thank you for watching Biology My Passion.